So hello, everyone. I would like to welcome you to today's webinar on advanced targeting sequencing, unraveling the complexity of plant genetic diversity. My name is Eva, and I'm Senior Marketing Specialist in Twist Bioscience, and today I will be your host. Before we start, I would like to give you a few housekeeping guidelines. First, all participant lines will be muted during the webinar. Please submit your question in the Q&A box, not the chat, and we will address all the questions after the presentation. If you include your name, we can follow up with you after the webinar in case your questions isn't answered live. After the presentation, we would appreciate if you could take 1 minute to complete a short feedback survey. So now I would like to introduce you to our speakers for today. Caroline Kalou works in a wet lab team, and William Marand is part of the bioinformatics team at the French National Research Institute for Agriculture, Food and the Environment, and the French Plant Genomic Research Center. They collaborate with public and private partners on projects focused on the characterization of whole genomes or targeted genomic regions across various plant species. And during this webinar today, we'll cover the following topics. We'll start by looking at how access to high-quality plant genome assemblies is helping us better understand species diversity. Next, we will dive into how differences within the same species can be studied using targeted enrichment. And then we will introduce 3 innovative strategies developed by the French Genomic Center to capture large, complex regions of plant genomes. Finally, we will show how this approach works using 2 examples, a pilot project on the 60 kilobase gene region linked to disease resistance in common bean, and a larger study on bread wheat targeting, a 750 kilobase region. We will end with the conclusions and take-home messages before taking any questions you may have. So thank you again. Enjoy our webinar. Caroline and William, the stage is yours.
Okay, so yeah. And the video?
All good.
Sorry, all good?
Yes, everything works perfectly.
Yeah, perfect. So we just add the video and try to go to the beginning again. I'm sorry, it just it doesn't come back to the beginning. Okay, now it should be okay. So sorry for this list of perturbation. Okay, so I will start first about I will present further our unit and the different projects we are involved in, and then Caroline will continue on the on Twist. So we are working at the French Plant Genomic Center. That is a unit service that is located in the southwest of France, and it was created 20 years ago by INRAE in order to support the plant genomic program and research. And at the beginning, we were mainly working on the plant back library. We were a depository center of the different genomic program in the world, the different consortium like the wheat genomic sequencing consortium, or the barley, or the tomato, etc. And then we had to produce, maintain, and distribute these tools. And but also in parallel, we were developing innovative approach for the production analysis of genomic data. And also our mission was also to develop collaborative project. And we had host and train, we used to host and train students and scientists. So in these 15 years, we developed several tools highlighted here. And we will focus on the first. I will talk a bit about our whole genome characterization project, and Caroline will talk about the capture. Just why a plant genomic center? Mainly because the plant genomes was known to be big and complex. And first, you can see in this table that the plant have large genome size, from small to large. But for example, the sunflower and the maize are around 3 gigs, like the human genome. But when you go to the different crops that we use to produce, like the barley or the wheat, it's much larger than the human genome. And also, one of the problem for the genomic, for the people that work on genomics, when you want to assemble a plant genome, is that it's full of repeated elements. For example, in the maize and in the wheat, you have more than 80% of repeated element. The other issue is that it's a polyploid. You have a lot of polyploid genomes in plants, and also when you compare two varieties, you have a lot of differences between the two genome sequence. So one reference genome is not always it's better to have several sequences. So during all this time, we have worked on a different project and mainly focused on two types of projects. The first, the targeted sequencing, or the whole genome sequencing. And I will start to discuss about this aspect. And because the scientific, the good starting point is the genome. A lot of times the researcher come in our lab and they want to have their genome. So we think that it was the best tool to start with their plant material and to give them at the end the whole sequence of their genome. And so for this purpose, we acquire all the expertise at every step. So if you send us seeds or plant leaves, we can grow them and then do the high molecular weight DNA extraction and then prepare the library for the sequencing, and then send it to the INRAE platform like Gentian. And after that, receive the data, assemble it, QC the assembly, and scaffold them, and now recently annotate them, the genome. So in 5 years, we were able to produce more than 200 genomes of high quality, and from more than 80 plant species and some few animals, because sometimes the people do not know how to extract their DNA from their lizard or aphids. And so they ask us, and okay, we're nice guys, so we can do it. And so we have done from small genome to big genome, from small to large genome, like the cypress, the cupressus that is around 10 gigabase, and also from homozygous to heterozygous genome, and from crops to trees and non-model plants. So a large variety of plants. And but sometimes you don't want to have the whole genome. You want to focus on your region of interest, and so it's why we have developed and worked a lot on targeted sequencing. At the beginning, it was more on a targeted back libraries construction and screening. So we were building the library and screen it and sequence your region of interest, several bags that covered your region of interest. And after that, when the sequencing technology evolved, we have also developed some approaches about large DNA fragment capture. And in that, so we are talking about region of interest. When we are talking about large, it means more than 1 kb. It means something bigger than a PCR. A typical PCR. So it's why it was important to develop this aspect, because nothing exists at this moment. And so when we are talking about that, we are in the middle here. No, I don't see my, yeah, maybe we can see here. Okay, we're in the middle between the high quality genome and the genotyping technologies. So we have tried different technologies like Simplix, adaptive sampling, and Twist, and I will talk more about it. It's not at this moment. It's not you want one region of interest. You don't want 1 or 2,000, but you want between 10 and 100 or 200 genomic region of interest from a panel of genotypes. And you want to limit the, if you go only to the target sequencing, it will limit the sequencing cost, also reduce the data volume, and simplify the assembly. So the three technology that for the capture that we will talk about is Simplix, Nanopore, and Twist. They all have their advantage and bias. And Simplix was quite nice because it only need one PCR, specific PCR marker. And then you can capture around 80 kb with only this information. So then Nanopore is quite good also because you don't need any, you don't need a specific machine except the sequencer, and you give the reference genome, and then you receive your sequence of interest. And at the end, you have the Twist, and you with Twist you can multiplex a lot of sample. You need a reference genome, and you can also capture a very large region of interest. So the two first, Simplix and Nanopore, are in progress in the lab, and Twist was validated and is now proposed by the CNRGV as a service. So quickly, how it works, the Simplix. So you have a specific marker. Okay, I don't think this way, but anyway, you have high molecular weight DNA. Then you create droplets in which you have the DNA and the PCR marker, and then you will sort the green droplet that are positive droplet in which you have your region of interest. And after that, you sort your green droplet and you sequence it. And you have a step of Phi29 amplification because you select only a few hundreds of molecules. So you need to amplify before the sequencing. So we have worked on it on the sunflower, and you can see here that we can produce a lot of data. We also have a very good enrichment, more than 2000x in your region of interest, in your gene of interest. And you can also have all reads for 80 kb. And so it works very well. But the problem is, during this process, you do a Phi29 amplification, and this amplification create chimeric reads that create different issue during the assembly. At the end, you have, instead of having 8 kb reads, you have smaller reads, and for the assembly of plants, it's not so good. So we still continue to work on this technology because the good point is that you need only few information at the beginning, and you obtain a very large region. And so we are working on the lab aspect and on the bioinformatics aspect. The second approach is the is adaptive sampling from Oxford Nanopore. And so the principle is very easy. You give to the sequencer your reference sequence, and if during the sequencing step, if the sequence corresponds to your reference, it will continue the sequencing. And if the sequence that is sequenced do not correspond to your reference sequence, it rejects the DNA. So at the end, you are supposed to obtain only a sequence that corresponds to your region of interest. So we have tried this approach on the common bean for a small genome with a region of with all the NLR gene of the genome that correspond to 2 megabases, and we obtain also a nice enrichment of the genes and you have a nice enrichment of all the sequence of interest. So on the theory, and practically, it's easy and it works well. But at the end, the enrichment is not so big because you have, instead of having 100 of gigabytes of data, you have 20 gigabytes of data. So all this accumulate together, make that the enrichment is not so high. But we are still trying this approach and see the efficiency and multiplex more and more sample to see where we can go. So now I will let Caroline talk about the different Twist projects.
So now we're moving on the third targeting sequencing approach, which was developed by Twist Bioscience. So you can access online Twist protocol for long-read library preparation and enrichment by following this link. And there is also an application note from PacBio, which describes the best practice by combining Twist capture with hi-fi sequencing. And finally, we develop our workflow for plant genomes based on these two resources. So I will detail how the capture protocol works. And just before to start, we need to apply to Twist bioinformatics team for a custom probe panel design specific to our target region. And we also need, at this step, reference genomes to minimize the off-target capture. So first, the process begins with a fragmentation of high molecular weight DNA, aiming for an average fragment size of 8 to 10 kb. And this size is optimal for amplification steps later in the workflow. It followed by fragments preparation and blocking step. And this step helps to prevent non-specific binding during hybridization. Then fragment DNA were hybridized with specific biotinylate probes, and the target capture were target fragment are captured using magnetic streptavidin beads. And after that, we release the captured DNA, perform a post-capture amplification, and proceed to long-read sequencing. So in our case, we use PacBio technology, but we can also use Oxford nanopore technology. And finally, data are assembled and analyzed. So I will illustrate how we apply it across 3 projects at CNRGV. So we first start with a pilot project on Common Bean, which has relatively small genomes of 550 megabases, and we target a region of 63 kb. And this project allows us to test and validate the protocol on 10 genotypes and using a panel of 90 probes. Next, we scale up to maize, so a more complex genome of 2.2 gigabases, and we target a large region of 270 kb split across 2 loci. And this project involves 16 genotypes and use over 2,800 probes. And finally, we applied the technology on bread wheat, so which have a massive genome of approximately 15 gigabases. And in this case, we target a region of 750 kb across 22 genotypes and using a panel of over 1,000 probes. So as you can see, the genome complexity, the target region size, and the sample number have progressively increased, and demonstrating the scalability of the Twist capture process for large and complex plant genomes. So now I will detail more each of these 3 projects. So first we start with the pilot project focusing on the 63 kb region on the Common Bean genome. So following the Twist capture workflow, we prepare a multiplex PacBio hi-fi library containing the 10 captured genotypes and sequences on the SQL2 system. And the run generates 4.7 gigabases with a read mean length and N50 quite similar, approximately of 6.5 KB. And this read length is consistent with the library size because you can see on the femtopus profile a mean peak of 7.3 KB. And this is also reflected by this plot, which is characteristic of hi-fi data and confirms that most of the reads fall within the expected size range. And also, additionally, this plot gave an overview of how much data is contributed by reads of different lengths. And despite the good quality of reads, sequencing productivity was lower than expected because we recover less than 5 gigabases compared to the typically 20-25 gigabases expected for this fragment size on the sequel to run. And this resulting and directly impact the sample yield between 400 and 500 megabases of hi-fi read per sample. From the raw sequencing data, first we extracted the reads mapping on the region of interest, and we recover between 4 and 5.2 megabases for each sample, and this corresponds to a 56 to 87x coverage. The read mean length and the N50 are very similar across the samples and in line with what we observe in the data sets, raw data set. And also we calculate an in silico enrichment ranging from 76 to 120x, and which is a strong indication of the efficiency of the capture. Finally, after that, we may perform a de novo assembly, and we obtain only one contig on the target region for 9 out of the 10 samples. So only one genotype required two contigs to fully span the region. If we have a look on the right, you can see the alignment of the alignment of the reads of each genotypes against the region of interest. And you can see that coverage is both high and uniform across all the samples. And we can note that for the genotype with the region of interest assembled 2 contigs, the breakpoint corresponds to a deepened coverage observed in all the genotype at this position. So even so, the sequencing yield was lower than expected, the capture was efficient, and finally we succeeded to obtain an assembly of the region of interest for each of the genotypes. But after observing the low sequencing productivity in our first experiment, we conducted some wet lab tests to improve this productivity. So here is the comparison between library from initial experiment and one library after optimization. And one of the most change that we made is introducing a cleanup step on capture library just before the sequencing library preparation. And with this improved library, we recover 5 to 6-fold increase hi-fi reads compared to the first experiment, and it's now matching with the initial expectation. Also, the read length is remained consistent, and finally, with this improvement, it allows us to reach up to around 2 gigabases. And this opens the door for more multiplexing samples per run. Due to the data quantity generated with the new library, with its improved library, we recover more reads mapping on the region of interest. So there is a direct impact the coverage up to 300x. So the assembly were consistent across both libraries, and with more data, we succeed to have single counting for all of the 10 samples. So finally, this cleanup step on the capture library is a critical factor for enhancing data production. Now we are moving on the second project, focused on maize. So that, as mentioned earlier, we target a larger region of interest of 270 kb. Like the Common Bean project, after Twist capture workflow, we prepare a multiplex PacBio iFile library, but in this case containing 16 capture maize genotypes. And now we sequence on the new PacBio system, so the Revio sequencer. So the run generates approximately 54 gigabases, and with a read with read min length and N50 quite similar that we obtain for the Common Bean project, and also consistent with the size of the library because we have a mean peak of 6.9 kb on the femtopulse profile. And the sample yield per sample was between 2.3 and 4.6 gigabases of iFi reads. So for each samples, we recover between 140 and 340 megabases of reads, mapping on the two targeted region with read min length and N50, similar across the samples and consistent with raw data sets. And this results in a very high enrichment because the coverage of the target region is between 500 and 1,200x. And based on this data, we perform a de novo assembly. So for each samples, the region of interest at each locus is assembled in approximately 3 countings with a total size ranging from 60 to 140 kb. And as you can see, the counting coverage is very heterogeneous, reflecting the complexity of the region. Here it's a visual example showing results for one genotype at one locus. So in gray, you have the coverage curves that represent the reads mapping to the region of interest. And as you can see, the reads are concentrated where the twist probes are, because twist probes are shown in blue. And here you have also the assemble counting from the de novo assembly. And just to have a look at this position, there is a highlight in orange. There is an important change involved in the studied phenotype. And due to the complexity of the region, there were few probes designed at this position, but we will be able to recover reads and to obtain a counting containing the gene of interest. And if we have a zoom on this gene, so here it's a dot alignment of its sequence between two nice genotypes. And as you can see, the alignment is disrupted by a clear gap in the middle of the plot, corresponding to a 4.5 kb insertion in gen 2 relative to the reference from genotype 1. So this example illustrates the power by combining target capture with a long read sequencing, because we can have more than only assembly on the region of interest, but allow to give structural variant detection. And finally, the third project highlights the application to a more and complex challenge in genome, so the breed width with a genome size of 15 gigabases. We focus on this project on 22 genotypes. So after the twist capture workflow, we divide the 22 samples into a multiplex PacBio iFi library, and each library is sequenced on the Revio systems. So both library produce comparable results, generating between 42 and 53 gigabases, but however, the read length is slightly shorter than expected because, for example, in library 1, The N50 read length is 4.9 kb, and you can see on the femtoples profile that the estimation of the library size is 6.3 kb. And so we sequence on the first library 13 genotypes and 9 on library 2, and the sample per yield was between 2 and 3 and 5.1 gigabases of hi-fi reads. So we, like the MAIS project, we recover between 140 and 430 megabases of reads mapping to our region of interest. And this is, we obtain a strong enrichment of target region with a coverage between 190 to 570x. And the in silico enrichment was estimated to 1,500x, and which is particularly impressive given the size of the genomes. So based on this result, one smart cell could have been sufficient to cover all over the 22 wheat genomes, indicating that higher level of multiplexing are possible in future experiments. So we perform a de novo assembly, and here is a result of what we compare the two maize genotype with the contrasting phenotype, and we focus on 6 genes known in the region of interest. As you can see, so genotype 1, the region of interest is assembled in 3 contigs instead of only 2 contigs for the genotype 2. And the difference between the 2 genotypes is the gene copy number variation, because if we have a look on the gen 2, gen 2 is present in 7 copies in genotype 2 instead of only 1 copy in genotype 1. And gen 3 clearly appears absent in genotype 1, but present in 3 copies in genotype 2. So this example illustrates also that the power of the combining target capture with long-read sequencing to detect gene copy number variation. And just to sum up, so now we have an efficient solution for targeting large region of interest, applicable across a wide range of genome complexity, from smaller genome like bean to highly repetitive ones like wheat. To compare to whole genome sequencing, the cost is significantly reduced, in particularly due to the ability to easy multiplex large sample sets. And one thing to be written also is that the prompt design is a really critical step in the workflow, and the success depends on that, because no prompts means no reads. And this approach requires a very low amount of input DNA, which is often a limiting factor for plants genomics. And finally, the use of long-read sequencing allows to generate accurate assemblies of targeted region due to the average read length of 6 kb. And which is especially useful for detecting structural variant and also gene copy number variation. So before closing, we would like to thank Twist Bioscience team for their support, and also to give us the opportunity to share our work today. We would like to thank our collaborator for working on their project, and also the Bioscience platform for sequencing our libraries on PacBio systems. And also, if you are interested to learning more or discussing for potential collaboration or just staying connected, feel free to reach out by email or just follow us on LinkedIn. And thanks everyone for your attention.